I'll introduce myself in the chat. I'll get it started. I'll say, oh, we have somebody here from Rayford, North Carolina. Very cool. And because we are recording, we may read out some of what you put in the chat to the world and say, if you have questions, they might get read out. So that's something to keep in mind, but we don't want to discourage you from being active in the chat and communicating with all of us. We have people from Atlanta, Georgia, the Medical College of Wisconsin, Milwaukee. Oh, very cool. Good representation from all around. All right, so let me get our slides going, share my screen. We'll go ahead and start presenting. So this is the Citizen Science and Library series. A big thank you to all of our partners who helped put this together today. It wouldn't have been possible without support from the network of the National Library of Medicine, All of Us Research Program, and the Scistarter team. I'm Caroline Nickerson. I'm a program manager from Scistarter. I have some of my other Scistarter colleagues on the line. And before we get into the nitty gritty of the event and our featured speaker from the Eterna Citizen Science Project, Do-Soon Kim, I'm going to pass the mic to Emily Hamstra from the network of the National Library of Medicine Pacific Northwest region to give a brief introduction. Emily?
Thanks so much, Caroline. My name is Emily Hamstra. I'm the Outreach Coordinator for the Network of the National Library of Medicine, Pacific Northwest region, and I'm one of the hosts for your webinar today. This webinar is the final webinar in the Citizen Science and Libraries series. Today's webinar, Help Develop RNA-Based Medicines, is brought to you by the Network of the National Library of Medicine, the All of Us Research Program, and Scistarter. I'm here with my co-host, Nancy Shin from the Network of the National Library of Medicine, Pacific Northwest region, and Caroline Nickerson from Scistarter, and also Michael Balkenhall from the All of Us Research Program, and our guest speaker, Do-Soon Kim from Eterna. Next slide, please. Today's talk is going to be structured into two parts. Part one, or the first hour of the webinar, is for anyone interested in hearing about the Citizen Science Project Eterna and asking questions. Part two, or the last 30 minutes of the webinar, is designed for librarians, educators, and community organizers, and will provide tips for engaging your community in citizen science projects. Everyone's welcome to stay for both parts of the webinar, and we're going to give a clear cue when we're transitioning from part one to part two, just in case you can't stay for part two. Before we dive into the presentation, I have a few more announcements. Next slide, please. Today's session is being recorded, and the recording will be posted to YouTube after the session. Closed captioning and ASL interpretation are available. The co-hosts will be monitoring the chat throughout the session, and please share any questions or comments in the chat box and share those to all participants. Questions will be collected and addressed during the Q&A session. Slides are available for the webinar upon request, and they're also going to be posted to the Scistarter site. If you want the slides via email, you can email those to me, and I'm going to put my email in the chat, or you can email me for the slides. Put my email in the chat there. If you're a librarian and you're interested in obtaining Medical Library Association continuing education credit for attending this session, you'll complete the evaluation that's provided at the very end of the session, or the end of part two, and we'll provide more information about that at the end of the session. And now I'm going to hand things over to Caroline Nickerson from Scistarter.
All right, thank you, Emily. And just a reminder, if you haven't yet, put your zip code in the chat, say hello. It helps us keep track of who we're reaching. And we'll be monitoring that chat throughout for any burning questions. So just keep that in mind as well. And for those of you who might be watching on YouTube, we're also monitoring that chat too. So don't be afraid to pop your questions in the YouTube comments as well. So we like to start things off with a poll, just to figure out, who we're in the room with or what level of experience you all have. And there are no wrong answers here. If you've never done citizen science before, that is A-okay. So yes, no, unsure. Have you participated in a citizen science project before? Get your votes in. Oh my gosh, it's about split. It looks the votes are coming in. I'm seeing about slight majority have participated before, but quite a few haven't, which is really exciting for me. I love getting new people in here who haven't done citizen science before. I'll give you all a few more seconds to vote. Get those votes in. Yes, no, unsure. Have you participated in a citizen science project before? And if you're watching on YouTube, feel free to give us your feedback in the YouTube comments. And I'm going to end the poll and share those results with you all. So it looks like the majority of you have, but there's quite a few of you who haven't, and that's totally fine. We're going to give you all the tools and all the info you need during this event to do citizen science, and in the breakout session to lead others in citizen science and specifically the Eterna project. So we'll stop sharing those results. Now, let's go to the next slide. Now we want to know a little bit about you. So are you an aspiring citizen scientist, a parent of an aspiring citizen scientist, a teacher, an educator, a troop leader, etc., a librarian or part of a library staff, a researcher or a project leader, just a bored person on the internet, or none of these? Pick the one that corresponds best, pick all that apply. We just want to know, who are you? Get those votes in. I'll give you a few more seconds to vote. It looks like we have a few aspiring citizen scientists, some parents. We got a few educators in the room with us, a few library staff members, some researchers. We do indeed have 2 bored people on the internet. I hope that this presentation entertains you and that you're not as bored afterward. And we have a few folks who are none of these. So I'll give you a few more seconds to vote. 3, 2, 1, and I'm going to end the poll. And I'll share the results. So it looks like the majority of you are library staff, but we do have quite a few researchers and educators. We have some citizen scientists, some parents, some bored people on the internet, and some people who are none of these. By the end of this, you will all be citizen scientists, and hopefully you'll have the tools to get others involved in meaningful research. So I'll stop sharing those results. All right, so citizen science, what is it? At Scistarter, we like to think of it as a collaboration between scientists and those of us who are curious, concerned, and motivated to make a difference. Basically, citizen science is any way you can volunteer to move science forward. That can be through collecting data or analyzing data. And there are thousands of citizen science projects listed on Scistarter for you to discover. People add their projects, events, and tools to Scistarter when they're seeking you to volunteer. So you can search for things that you're interested in. Like if you're really into astronomy, you can find astronomy projects. If you want to monitor water quality, you can find water quality monitoring projects. Basically, citizen science is as broad as science itself. And I promise you that there's a project out there that will ignite your interests and help you make a difference. And today you're going to be learning about a really special project called Eterna that you can do online from anywhere in the world to help build RNA-based medicines and even vaccines. So we'll hear a little bit more on that in a sec. We recommend that you get started on scistarter.org/NLM. At this point in the presentation, I always tell folks, minimize me. Go ahead and open up a window and type this URL in. I'm also going to put it in the chat. Out of all the projects on Scistarter, we spotlighted these in partnership with the National Library of Medicine because we thought these would be a great fit to get you started. And believe it or not, you can make your Scistarter account and access other resources all on this page, all in one place, including the recording of today's event. So go ahead, go to this page, make your Scistarter account, and get started. Also on this page is the Introduction to Citizen Science tutorial. It tells you the what, the how, the who, and the why of citizen science. It takes about 30 minutes to click through. It's interactive. There are quizzes. There's a certificate at the end. There are videos. It's a lot of fun. And it's also offered in English and in Spanish. So for those of you on the line with us today who are community leaders, this tutorial might be really helpful for you to introduce your community to citizen science. And it was also written at a 5th grade level, so it could be accessible to all ages and all types of educational backgrounds. And even if you know everything about citizen science, I hope this tutorial teaches you something new. I think a lot of different people can get something out of it. So that's on the scistarter.org/NLM page for you to discover. And now I'm so delighted to introduce Joo-Soon Kim from the Eterna project. He's going to tell you a little bit more about him, because we always like to put that human face on these amazing research projects. And then he's going to talk about what Eterna's been up to with vaccines and how you can get started. So I'm going to go ahead and give control of the screen to Joo-Soon.
Hi, I hope everyone can hear me.
Yep, we can hear you.
Is my volume okay?
Yeah, you
okay,
sound
great.
good.
So first of all, thank you so much for having me to Scistarter. Thank you for Heather and also Michelle, who will be translating everything I'm saying to sign language, which I think is so wonderful. Because the spirit of citizen science, I think, is to make science accessible to everyone. And I hope that I'm able to share that with you and how we have tried to achieve that on Eterna, and how we're empowering citizen scientists to invent medicines for the next generation. So moving right along, I'll quickly start with the intro, like Caroline mentioned. How did I get here? I think this is a very interesting question for any citizen science effort. And for many people, it's a long and winding road, and you don't necessarily have to come from this very sort of rigid and very sort of pre-planned path. I think people come from all over, and I just want to show that off, that I also just took a long winding path to get where I am. So I did research in at university studying chemical engineering, which is nothing related to what I do now, but I started doing research in a field called synthetic biology, which aims to harness the power of biology for technological advances. And then I moved on to Northwestern University, and I'm actually currently in Chicago, so my zip is somewhere in the 600s, to study chemical and biological engineering, a little bit closer to what I do now. And I worked on RNA as a molecule and machines that are made of this biomolecule named RNA. And then through my graduate work during my PhD, I became involved in Eterna, which was this video game citizen science project that aimed to empower citizen scientists to design medicines. And at first glance, it sounded a little bit like science fiction. I didn't actually believe it was that real. And then as I started to get involved, I understood the power of citizen science and how wonderful it can be. So that's how I ended up where I am. And then recently in March, starting this March, I became very heavily involved in Eterna to lead a initiative called the Open Vaccine Project to focus on designing mRNA therapeutics and vaccines against COVID-19. So now that position is at Stanford University, where Eterna is run out of. So before I start on describing Eterna and what we're doing, I thought it'd be good to share a short primer on mRNA vaccines. It's all the rage currently. It's in the news all over for being the first approved vaccine against COVID-19 and the virus which causes COVID-19, SARS-CoV-2. But I wanted to sort of level the playing field a little bit to explain why mRNA vaccines and why all of a sudden they might be relevant and important. For those of you who are very formally trained in science, this may seem rather maybe high level, but I'm hoping that I'm able to give a quick overview of why they are relevant for those who may not be as familiar. And then second, I want to show you how citizen scientists can help in designing improved mRNA vaccines and how you can contribute to a research effort that is scientifically incredibly complicated and complex, but I'll show you the power of human intuition and citizen science in our efforts. So in short, I'm going to try to walk you through how vaccines work and why specifically mRNA vaccines have been important. So the first point that I want to drive home is that a vaccine is a molecule that trains your immune system to recognize and fight viruses and infections. So in SARS-CoV-2, what you want to do is you want to introduce a medicine into your body before you are infected with the virus that is going to teach your body to fight the virus upon infection. It's a lot like studying for a test. If you're given a test without studying for it at all, it's going to be really, really difficult, with the exception of very good test takers, I suppose. But you're sort of preparing your body to fight a virus in the case that you are infected. The way this happens is we want to teach your body what the virus might look like, but you want to teach your body what the virus looks like without the bits that make you sick. So on the coronavirus, so the word corona is actually derived from the appearance of the virus, as I've shared in the photo here. There are these three sort of popular proteins that exist on the outside of this virus. And the one that everyone has been talking about, and a great way for your body to learn what a coronavirus might look like, is to give it this S protein or spike protein that covers the outside of this virus. So what you're going to do is you're going to give your body a little sample of this spike protein, and your body's immune system is going to say, okay, here's a little protein that my body normally doesn't make. So if I see it, I now know what it looks like, and I know how to induce an immune reaction against it. So that's the sort of the very high level concept of how a vaccine against the coronavirus might work. It turns out that making the spike protein, which is the final, what we call immunogenic molecule, or a molecule that's going to induce an immune response, making it separately in a chemical reaction can be difficult. And in this case, it can be a slow process. So for other diseases that have maybe sort of been longstanding, this process of making a protein might have been slow, but maybe not as urgent. But for the coronavirus, that is a raging pandemic right now, especially in the US, that speed is really, really important. And how quickly you can get to this protein is really, really important. And this is where mRNA vaccines come in. So the way how an mRNA vaccine works is that instead of giving you the pre-made protein as a vaccine, which we typically do for things like the flu, what we can do is we can give your cells the mRNA message. And this image that I'm showing is called the central dogma of biology that you might have learned in primary school, where DNA goes to RNA, goes to protein. So we know that it may take a lot of time to make the protein bit. So what we're going to do is give your body the instructions of how to make that protein yourself. So we are giving your body the mRNA molecule, and then trusting that your immune cells will turn that mRNA molecule into the spike protein. And then from that spike protein that is made inside of your cells, it'll learn what the virus looks like, and thus have immunity against the virus in the case of infection. And I'm happy to take questions on what we do on Eterna, but also specifically mRNA vaccines, and especially for COVID. And I will try to field those as I see them. So I see we have a question in the chat. The question is, how does the cell recognize that the spike protein is a foreign protein that needs to be attacked? So if you're an immune cell, that's sort of your job, is to monitor foreign bodies that are introduced to you and to develop immune responses against it. This is not unlike if you get a bacterial infection or other infections like the common cold or the flu, how your immune system might react. Your body has a very good idea of what cells already exist in your body versus what cells are foreign, based on a lot of different complex biologies, but it actually has something called memory against cells that it's used to seeing versus cells it's used to not seeing. So because the human body doesn't make a spike protein on its own, your body has a very good idea of what is native to you and what is not native to you. And also there's a Q&A at the end. I'll try to come back to some of these questions if I don't get to them during the talk. And the question then becomes, why do we want to engage citizen scientists, and why are they such an important part of helping designing improved mRNA vaccines? So if you sort of get into nitty-gritties of how many different ways can you code for a spike protein, it's a pretty large protein, and turns out that there are 10 to the 600 ways for us to make an mRNA message that codes for a spike protein. And just to give you a reference of just how ridiculously large that number is, there's only about 10 to the power of 11 stars, they estimate, in the Milky Way galaxy. And there's only about 10 to the 13th cells in a human body, right? So you can imagine the number of possibilities that we would have to sift through to figure out which mRNA machine is the best way to encode for the spike protein is enormous. And it's not something that we can explore exhaustively, meaning we go through every possibility and test them individually, especially when you need to do it quickly. So what we do on Eterna what we did in Eterna, we mobilized the community, which includes thousands of video game players, to design better mRNA sequences that code for the spike protein, which can be used as a vaccine. And we do this through crowdsourcing in video games. And I'll sort of share with you what Eterna is and how we leverage that platform towards open vaccines. So this is how Eterna works. It's a website. You go and make an account if you're over the age of 13. You'll have a set of tutorials that'll teach you to manipulate sequences of RNA, and you'll be shown what it looks like in real time. And then after you submit a design for an RNA that you've made, we allow the players to vote on which RNA shapes or sequences they like best. So this is totally democratic, and whoever gets the most votes gets to win the voting stage. And this is the sort of the secret sauce of Eterna, is it doesn't end once a design wins on the voting platform. What we do is we actually get those sequences and we synthesize them in laboratories at Stanford University, and we subject those RNAs to cutting-edge biochemistry experiments done by expert research scientists and a wonderful team at Stanford University who's in charge of that. And then what we do is we return the results of our scientific experiments back to the players. And sometimes that means simplifying the results a bit, but oftentimes it means through this process, teaching the players the science that they need to learn to interpret these results. And I'll share with you soon, this sort of allows people to learn science at the very cutting edge research level and has led to peer-reviewed publications by citizen scientists. So, and then we can go around this loop multiple times. This process of players designing, voting on the best designs, us doing physical experiments and returning the results. And this is an abstraction, or this is a practice of the scientific method where you form a hypothesis in your design, you then test it, and then you refine your hypotheses and designs based on the results. And as I mentioned briefly before, we've sort of tackled many different problems in RNA medicines and not just mRNA designs. And then they have resulted in many scientific publications. So 3 of sort of the ongoing efforts that we're focused on outside of our vaccine work are thermodynamically perfect riboswitches. So the application for these are RNA medicines that can be controlled very precisely. Because one of the worries that people have about therapeutics is once a molecule is in your body, how do you make sure that the reaction to it might not be too harsh? So the one way you can do that are through these little RNA machines called riboswitches. And Eterna is one of the best platforms where video game players have been able to design many different perfect designs for these things. Another thing that we're incorporating into our platform is rational 3-dimensional design. So getting away from just what we call the 2D or secondary structure of RNA and abstracting that out to what the RNA looks like in 3D for players to start to imagine on different dimensions what they might be designing. Another exciting program we have is called Open CRISPR. CRISPR might be a technology that you're familiar with. CRISPR relies on a very key RNA molecule called the guide RNA. And we have efforts to design a very safe and equitable guide RNA molecule such that it can be controlled very exquisitely in the event that we don't want the genetic editing process that CRISPR does induce in the human cell. And that has led to over 20 publications, some which I think is really, really cool, who have authors who are only citizen scientists. So on the author list, you might find people like me, a career professional researcher, authoring publications in a professional setting. But what we found is that citizen scientists through Eterna have learned so much science that can be published in a peer-reviewed scientific journal that they can actually write the paper themselves, do all the analysis, and they have essentially become on par with very good graduate students and even professors through their efforts on Eterna and through learning science through learning RNA biochemistry through citizen science. I wanted to share with you, if this works, a quick demo of what Eterna looks like when you actually play it. I don't know that I hope
we can actually I can take control back and play it for you on our Google Slides. Let me do that
really sounds quickly. Great, yeah. And I'll sort of narrate what's happening on Eterna so you can see in a quick, I think it's about a minute, what's happening on Eterna if when you play the video game.
All right, I'm going to share my screen again, and let me just get it up really fast.
Sure.
And I've seen questions pour in, so everyone, we're about to get to the Q&A section, so keep those questions coming. I promise we'll answer them. Okay, sharing my screen and present and play.
Cool. So this is what a puzzle looks like when you start it on Eterna. The goal here is to design an RNA molecule that looks like a pair of glasses. So this is probably not a real RNA molecule that's found in the body, but players make artistic puzzles like this all the time. And what this puzzle tries to do is teach a concept called base pairing, that certain bases of RNA, which is made of 4 different bases, A, G, U, and C, we want to teach the concept that certain letters of RNA pair with other letters of RNA, just like it does in DNA. So what we try to teach the player to do is sort of iterate back and forth between the sequence and what it looks like to teach these design rules that G's pair to C's, or green bases pair to red bases, or A's pair to U's, or yellow bases pair to blue bases. So that's what an Eterna puzzle looks like. This is a very simplistic case, of course. But these puzzles build upon the complexity of these simple design rules, and then players build upon those even more to develop strategies and further very complex rules for designing RNA, which have stumped professional scientists for now a couple of decades. And then finally, I just wanted to share that the biggest thing about Eterna, I think, is that it's incredibly accessible. If you have a web browser and an internet connection, we're so happy to see new players. We're really excited to engage with them. You can engage with us through the standard social media platform, and also the campaign that we have currently going is called the Open Vaccine Campaign against COVID-19. We also have mobile apps that we developed in this pandemic for to be more accommodating to people who like to play games on their phone instead of the computer. So we have now the app open on both Google Play and iOS for iPhone users. And yeah, thanks so much for having me. I'm happy to take more questions, and I'll be back at the Q&A period, I think.
Yeah, we're going right into it. So Nancy from NNLM PNR is going to kick us off with some questions. And I noticed a few came in. So while Nancy asks her first questions, everyone else, please get your questions in the chat or in the Q&A function for due soon. But I'll pass the mic to Nancy now to ask those questions.
Okay, thanks, Caroline. Welcome to our Q&A period. My name is Nancy Shen, and as you may recall, a week from today, you received an email from me asking you to brainstorm some questions to ask Doosoon during this Q&A period. So based on that feedback we received from you, we have curated a list of questions to ask Doosoon about his exciting work. Before we do that, I noticed that some questions came in through the chat, so we'll address those first before we go over the curated list. So the first question that they asked Doosoon is the RNA molecule wrapped in a protein in the vaccine?
So it depends on the vaccine. It's not publicly disclosed, to my understanding, what exactly it is wrapped in. But to my understanding, the 2 vaccines that are approved right now, one from Pfizer and one from Eterna, are not wrapped in proteins, but rather formulations of chemicals, often called lipid nanoparticles, that help deliver the RNA molecules to the right cells. So not proteins, but synthetic chemical formulations used to sort of wrap the mRNA like you described.
Okay, awesome. And the second question from the chat box asks, is it true that Pfizer designed its vaccine 2 days after China gave its genome sequence in January?
I couldn't speak for Pfizer, but I do know that designs for the mRNA vaccines or at least candidate designs for the mRNA vaccines were decided on, I think, within a week of the genome sequences being released. And that was one of the advantages of mRNA vaccines, is that you can design a vaccine very, very quickly in terms of the mRNA sequence soon after the genome sequence for the virus was determined. And I should say that there's a lot of reasons for that speed. One reason is that mRNA vaccines are fundamentally very fast platforms. Another reason we were able to do that is because there are decades of research on other viruses that feature spike proteins. So the US was not as affected, but in 2003, there was an outbreak of SARS in China and East Asia. So we learned a lot from previous outbreaks of coronavirus-induced infectious disease pandemics that we were able to apply to this SARS-CoV-2 coronavirus that allowed the designs to proceed so quickly.
Actually, I'll jump in with a quick question there. So what's different with the vaccines that citizen scientists are helping you design with Eterna versus the ones that we're hearing about in the news right now with like Pfizer and Moderna?
Yeah, so we actually honed in on a very specific problem or a very specific challenge that, if solved, would make the vaccine way easier to distribute and to vaccinate more people. So Eterna, being a citizen science platform, one of our core values is that medicines should be democratized and be very equitable, both in its development and also in distribution. One big scientific challenge right now, or maybe an engineering challenge, I should say, the current mRNA vaccines that you might have read in the news, is that they have to be transported very, very carefully in very cold conditions. And it's because mRNAs have this innate characteristic that if left out in warm temperatures over time, they will spontaneously break apart. And once it breaks apart, it's not very useful. So there's this challenge of making very stable mRNA vaccines, because we on Eterna realized that having a vaccine that requires a lot of really advanced technology for supply chain may limit who are able and eligible to get the vaccine, typically to wealthier, more technologically advanced areas. So we recognized on Eterna that if we wanted a vaccine that will truly go out to developing parts of the world and immunize a wide swath of people, we had to make it very stable against thermal degradation. So what Eterna does is design mRNAs that are specifically resistant to thermal degradation so that we can ship more vaccines to many different areas and people.
Got it. And before I let Nancy get back to our regularly scheduled programming, I also have another quick question. I scanned the attendance list. I noticed that quite a few people who are with us are K through 12 educators and children's librarians. Who does Eterna and how young could they get? Like, what level of student would be able to participate? Because I know a lot of adults do it, but have you seen students do it successfully?
Yeah, so we do have a minimum age, which is 13 to participate in Eterna. What we're pushing as of late, and I added this to a slide later, we're making a big push to incorporate Eterna into educational curricula, because we saw an opportunity, especially with remote learning, that any way to make schooling more fun might be really beneficial, especially for kids who are stuck at home and maybe kids who like playing games. So we saw this opportunity to make Eterna more education friendly through an initiative that we launched this summer called Eterna Rise. And we've incorporated it into a college curriculum at a historically Black college and university in Houston. And we're reaching out further to make the website more friendly to younger students. So I would say typically any student in middle and high school over the age of 13 that meets our age limit should be able to get going on Eterna very, very quickly. There's not a lot of technical lingo that we use on the site on purpose to make it as easily accessible to younger students or people who may not have formal training in science.
Got it. More on that in the leadership breakout group, everybody. Get excited for that at the end of the event. I'll let Nancy get back to our main questions.
Okay, thanks, Caroline, for that needed break there from the question period. But going back to the chat box questions, one person asked, would there be an advantage to porting these games to VR, which I assume is virtual reality, to incorporate 3D?
I think if that's possible, that would be amazing, because I think that is sort of the future of three-dimensional games. And it would really add another dimension to how we think about the RNA folding problem, which is the fundamental scientific problem that we try to teach on Eterna. So yeah, I think there would be a huge advantage to porting it to VR, and it would really allow especially younger students to interact with it in a very unique and advantageous way.
Okay, great. And I have another question that asks, if some range of viral load is safe, then is it okay to assume that humans might be carrying viruses for unknown diseases?
That is a question in immunology that I'm not qualified to answer, but it is very possible that humans are carrying viruses that are, say, not non-pathogenic. So for example, in your gut, you have a gut microbiome, which are a collection of bacteria. There are viruses that infect bacteria. So it is very possible that you might have some virus in your gut and it just doesn't cause a disease. That's certainly possible, doesn't seem out of the realm of realities. But an immunologist or a virologist would know this much, much better. Specifically for SARS-CoV-2, I think that is one of the challenges, is that you can carry the virus without being sick. But again, it's not my area of expertise, so I'm hesitant to answer it too, certainly.
Okay, great. Thanks, Susun. So now we'll address the curated list of questions that I got from people through email. The first question asks, what strategies have you used to tell the story of Eterna?
What strategies? I don't really have an explicit strategy I use. I think so the one thing I try to do is really focus on the power of the Eterna community. I think that makes the story very easy to tell. If I focus on the citizen scientists that are involved in Eterna and all the interactions I've had with them. So for example, we have weekly town halls where the scientific people and the game developers and also project leaders like myself will just get into now a Zoom room and talk with the citizen scientists to answer questions about science, to ask them questions about how it's going and what the video game features that we might be able to use. And I think a lot of those interactions over time really make it easy for me to tell the story of the citizen scientist and not necessarily just what the science is doing or just what the research output might be. So I try to focus on the people who play Eterna, who really make it the community that it is.
Okay, great. We have a lot of librarians here today, so this question might interest them. Mm-hmm. How have libraries been a part of Eterna?
So I think for me, this has also been a learning experience and learning what all that a library could do. So when I was very young, a library was somewhere you went to go get a book, right? But actually, libraries, I think, have really transformed into a sort of a dissemination of knowledge. So I think librarians, especially those who are very well connected in the community, have a really good idea of how it's sort of the gateway into the community of people who are intellectually curious. So I think by reaching librarians and allowing librarians to sort of disseminate the excitement or informing people in their community about options that are available to enrich their intellectual curiosities has been really, really beneficial and also very rewarding from Eterna.
I see another question in the chat, Nancy, that came in.
I see it right now.
Yeah.
Okay. There is a question in the chat that asks, is there a way for students to work with data they've generated on the Eterna platform? If possible, how does that work?
Yes, there is absolutely. So one tenet of Eterna is that we release all the data we generate publicly so that it is accessible by any Eterna player. So in terms of how would they work with the data, we have built-in data browsers that allow students to mine the data, meaning you can download it and you can work with that data set however you like. So it is open for you to take and use for educational purposes freely. If you're thinking about like tools that we can develop to better help students learn data analysis through the data that's generated in Eterna, that blends in well with the initiative Eterna Rise that I talked about, integrating Eterna into the sort of the scientific curricula at universities. And I'm sure we could find a level of statistical analysis that might be really well fit for primary school. So yeah, I'd be happy to explore that. It's something that we thought we've thought very deeply about, because a huge part of the scientific process is data analysis.
Okay, great. Thanks, Jason. The next question on our curated list is, what's something about Eterna that has surprised you?
I think the scientific rigor of citizen scientists. And I mean that in two ways. The first is everyone in Eterna, I can confident, I'll disclose, my Eterna rank is like 3,000, which means I'm someone who has just received a doctorate on designing RNA machines. And there are 3,000 people who play a video game who are better at designing RNA machines than I am. Right. So first, it's a humbling experience to know just how good video game players can get at a very scientifically challenging problem. And then the second thing that's really surprised me is just how curious people from non-scientific backgrounds are about science. So I thankfully I happened to get into science at a young age, developed a curiosity, and now continued on my scientific training. So to me, it was always been like a professional interest to some degree. But a lot of players, the top players in Eterna, some are accountants, some are botanists, like people without what we would consider the traditional STEM field. Their curiosities to learn about science and speak the language and be able to sort of not only understand it, but push that field forward has been super surprising, just how passionate people are and just how willing to learn about new things.
Wow, that's really amazing. And our last question from our curated list for today is, what's something you wished everyone knew about citizen science?
One thing I wish everyone knew about citizen science is how accessible it is and how friendly the community is. So it seems intimidating, I think. I think science in general could do a better job of being more accessible. So citizen science similarly can seem intimidating to someone who perhaps is not sure that they can make an impact in the platform, or not sure whether they can take their children to this community and if their children would thrive in it. But I would say almost every citizen science community I've interacted with, this sort of the common denominator in all these communities is how welcoming they are and how patient people are with your questions. So I think knowing that you should just jump into it and try it out and see if you like it. And if you don't like it, you can try a different citizen science platform. But I think that how low the barrier of entry is and how friendly the community is to newcomers is something that I wish more people knew about.
That's great. And thank you so much, Nancy, for facilitating those questions. I think we have time for one more question in this section before we move on to like the closing of our public program. But if you all have additional questions, we do have a leadership breakout group for 30 minutes after the main program. So starting at 3 o'clock Eastern. So you all will be able to ask your questions then for sure. We got a question in the chat. Someone asked, what other platforms are great for citizen scientists? So you can discover thousands of projects on Scistarter and even more health-based projects on the scistarter.org/NLM page. But I'm curious for Do Soon, are there other citizen science projects, especially ones outside of the health field, that you've participated in, just as a citizen scientist yourself?
A very long time ago, there was one where you took pictures of plants on your hikes and stuff, and then you send it off to be identified, that I really enjoyed. The name is escaping me, but that was probably my first. Yes, so that was like my first interaction with citizen science. And that was honestly one of my first interactions with technology. So I love that as a simple and easy way to get into science, of course, because there are very good botanists on the other end, plant biologists on the other end, but also it was just a fun thing because it pushed me to get outside and to integrate science into what was a very relaxing hike, a very fun activity.
And fun fact, iNaturalist and Eterna are both featured projects in the Girl Scouts journey on Scistarter. So if you're a leader or a volunteer with a Girl Scouts troop, you could conceivably have your students and your Girl Scouts do Eterna for a while and then go outside and do iNaturalist on a hike. Like these could be complementary activities, just something to keep in mind and maybe something we can dive into during the leadership breakout. So thank you so much, Dusun, for sharing this knowledge with us. I learned a ton, and thank you to Nancy for facilitating. We'll move on into our All of Us research portion. All of us supported this event and helped make it possible. So we'll pass the mic to Michael Blakenhall to tell us a little bit more about this exciting precision medicine initiative. Michael?
Hey everyone, thanks so much, Caroline, for passing it off. My name is Michael Balkenhall, and I work for the network of the National Library of Medicine. As Caroline mentioned, today's webinar is brought to you in part by the network of the National Library of Medicine, also known as the NNLM. And we are a program of the National Library of Medicine. And this is also sponsored by the All of Us Research Program. The mission of All of Us is to, the All of Us Research Program rather, is to accelerate health research and medical breakthroughs, enabling individualized prevention, treatment, and care for all of us. The goal of the program is to help researchers understand more about why people get sick or stay healthy. By looking for patterns, researchers might learn more about what affects people's health. The All of Us Research Program aims to build one of the largest, most diverse data sets of its kind for health research, with 1 million or more volunteers nationwide who will sign up to share their health information over time. The goals of All of Us and the National Library of Medicine partnership are to increase the capacity of public library and community-based organization staff to improve health literacy, digital literacy, and participation in scientific research, much like citizen science. The National Library of Medicine and All of Us have prioritized citizen science as a focus for engagement activities with and through libraries and other community-based organizations. You can go to the next slide. So if you're new to the idea of precision medicine, it's just one way that researchers are looking to advance the ability to serve the public health needs of all people. So All of Us is a precision medicine study, which essentially is a new way of looking at health treatment and prevention as being designed for individuals. This way, doctors can take into account a person's lifestyle, environment, and biological makeup to figure out the best treatment for that individual. On this next slide, we have some examples. The neat thing is that we already use precision medicine in so many different ways, such as myself right now with prescription eyeglasses. Eye doctors can tell us exactly which lenses will work best for us, but when we visit our regular doctor, they may be more likely to prescribe the same treatment or prevention that works for most people and not just us, or not just the individual. There's when a lot of people pitch in to help with this research program, we will build a large national database to eliminate one-size-fits-all medicine. Next slide. So crowdsourcing and citizen science is an NLM, a National Library of Medicine, an NIH priority topic for engagement and outreach. Through citizen science and crowdsourcing, the NNLM hopes to engage communities in addressing societal needs and accelerating biomedical science, technology, and innovation. We began this work with our partnership with Scistarter to create an online tutorial, which if you haven't seen it, you should definitely check it out, on how to run citizen science programs on your own. We did this to provide opportunities to engage in citizen science activities in and through libraries, kind of like this webinar, and as well as choosing curated citizen science projects that align with All of Us engagement priority areas. The National Library of Medicine and All of Us are pleased to provide this opportunity for you to engage and learn more about citizen science. And we hope that you stay actively involved with us and keep an eye out and maybe partner with us for some future events as we continue our work with Scistarter for an amazing program series that we're gearing up for Citizen Science Month in April 2021. So a little bit, again, to learn a little bit more about All of Us, maybe learn about a little bit deeper into the program, or get some questions that you might have answered, please do visit joinallofus.org/NLM, and we'll drop that into the chat too if you want to take a look. And I'm going to pass it back to Caroline.
Great. Thank you so much, Michael. Yes,
For sure.
please everyone go to joinallofus.org/NLM, and because all roads lead to Rome, we also have that link on our Scistarter.org/NLM page where you can access instructions for Eterna, the recording of today's event, and so much more. So thank you so much. So yeah, that's our call to action here. We want you to go to this page, participate in health-related citizen science, join the All of Us research program, even join us for Citizen Science Month in April 2021. I know we have a lot of leaders on this call, educators, leaders of community-based organizations, librarians and library staff. You all can plan Citizen Science Month events or even just get your communities doing these projects during Citizen Science Month and before it too. Wouldn't that be amazing? All of you getting everyone to do Eterna, keeping track of everyone's participation, really assessing your impact. I think that could be really cool. So that's my call to action for all of you. Please get out there and start doing some citizen science. And we also really want you to do this survey. It helps us learn how we're doing. And I'm putting it here because I know some people are probably not going to stick around for the leadership breakout group we have up next. But this survey is for everybody to just give us feedback about what you want to see in future events. If there are things that you really like that you wouldn't want us to drop, you can tell us that too. I'm putting that link in the chat, and you can go ahead and click that, or you can type it into your browser. So please fill out that survey. I think we're going to roll right into the leadership breakout group. So for those of you who are members of the general public who don't want to stick around and learn how to lead citizen science, that's totally fine. But basically, anyone who thinks they're a leader is a leader. You could get your church group doing citizen science. You could get your, for students, you could get your friends doing it. So I think anybody can learn something from this, but especially leaders in libraries and CBOs and educators will get something out of this next segment. So I'm going to pass the mic back to Emily from NNLM P&R to talk about facilitating public programs in libraries and community-based organizations, also known as CBOs. Emily?
Thank you, Caroline. So before I get started, I'm going to put a resource in the chat. My internet is running a little bit slow, so if there are any issues in my connectivity, please refer to that resource that I've put in the chat. So when you're planning a citizen science program in a library or a community organization, there are lots of different things to consider. And I have a few things on this slide to get you started. So first, think about the audience. Who's going to come to this program? When you're planning, consider partnering with maybe local organizations, schools, libraries, senior centers. Partnering can increase community buy-in and attendance, and depending on who you're partnering with, it might also help you share some of the costs or the planning of the program. Next, think about technology. So what's required to participate in the citizen science project? Do you need internet access, a mobile device, or something else? And does your audience have access to the technology that they need? If they don't have access, are you able to provide them with access to that technology during the program? Funding can always be a challenge. I encourage you to look to the network of the National Library of Medicine for possible grant opportunities for health-related programming in libraries and community organizations. Also think about the time and location. So what time of day are you going to host your program? Is it going to be a one-time program, or is it going to be a series of programs? Is it going to be an in-person program, or are you going to plan it as a virtual program? Also think about the marketing. So when you're planning the event, how are you going to get the word out? And I highly recommend checking out Scistarter's Library and Community Guide to Citizen Science, and I put that link in the chat. That provides you with more tips and information about citizen science programming. During the event, pay attention to who is at the event and ask them how they heard about the event. Also pay close attention to what types of questions attendees are asking. So if you're hosting a program on Eterna, are people asking questions about mRNA and vaccines, or are they asking questions about citizen science more broadly? And listening closely to those questions can help you inform your future programming. Maybe you want to do more programming in citizen science, or maybe you want to do more programming related to to COVID-19 and vaccination efforts. I recommend providing a handout with additional resources and information so that each participant leaves with some next steps in mind to continue their learning. After the event, take some time to reflect on what was successful and what made it successful, and also what you might want to do differently next time. All right, and I think it's back to you, Caroline.
Yeah, and I wanted to tell everybody, I'm putting this in the chat too, Emily really practices what she preaches, because she made a handout for today's event, which you can find on the NLM page. So I put scistarter.org/NLM in the chat, and that library resources page. Remember, you don't have to be with a library to use things on the library resources page, including our new hot off the press Library and Community Guide to Citizen Science. It's about 60 pages of citizen science goodness, walking you through everything you'd need to facilitate your own citizen science program, be it something that you do in person or virtually or some mixture of the two. But we're in a virtual world at the moment. I think everybody's been doing online programs, so I wanted to let you know that you can do it too. You don't have to be scared. I know that at the beginning of, when we started social distancing a little bit more and responding to new public health guidelines, a lot of the folks I worked with all around the world from different libraries, community groups, and other entities, they were really nervous about doing online programming. But I told them, as long as you acknowledge that you're not perfect, it's okay. I mean, I just made a mistake. It took me 3 tries to correctly type in the library resources link in the chat. So if you let everyone know that you're a human, but you're all there for the same goal, which is to make the world a better place by doing scientific research, then your program's already off to a good start and it's probably going to be a success. But that being said, do your best to have your online setup. If you have certain barriers, like let's say that your bandwidth is lower or you're having issues like staying connected on the Zoom, you could see if you can get some grant funding or support for like a Wi-Fi booster, for example. Something simple like that can make all the difference. And if you just have to be responsive to the issues you might encounter in a program. So there have been times where my internet bandwidth wasn't the best. I remember one time the internet went out for my entire city right before a really big program, but I was able to call in via phone, and my buddy Robin, who's actually on the line, was able to share my slides and walk through it while I explained them. That was teamwork right there. So the buddy system is key. You want partners and friends to help you through these events. And Scistarter is happy to play that role for you, especially going into Citizen Science Month. We're hoping to do hundreds of different events with groups all around the country, and your group could be one of those. If you go to citizensciencemonth.org, I'll put that link in the chat, you can access resources about how to book Scistarter's time so we can be your buddy for these Zoom events, and how to, enact these things yourself. But the buddy system is key. Someone like Robin's able to look at the chat and, add her own commentary or make sure I don't miss any questions. And same with Nancy and Emily, right? We were all kind of a team in this effort today to make sure that we were responsive to your Q&A questions and responding to you as viewers. And then for presentation material, I love a good shared slide deck. We all made this slide deck together. And I thought that was really helpful in understanding how the program would go. And live demos, they can be technically challenging, but they're really rewarding. So if you're able to, maybe if you have a smartphone, download the Zoom app on your smartphone and take people outside and do an outdoor project with them, or share your screen and participate in a project like Eterna. It can be a lot of fun to actually accomplish real scientific research, citizen science, in real time with the community that you're engaging. And I wanted to give you all an immediate example. The county of San Luis Obispo in California is doing an event with us right after this. They went through the process. They did an event with us over summer, and they had such a blast. They wanted to jump back in and do another one. So they went through the citizensciencemonth.org process of booking an event. They emailed back and forth with me a little bit about like what project they really wanted to hone in on, which was Globe Observer Clouds, one of the really cool NASA projects that helps you ground truth satellite data to help NASA understand if their satellites are working right from above and below, understanding the sky. And if you want to attend it, you're more than welcome, so you can kind of see this in action, everything that we've been telling you about facilitating public programs in citizen science. So the link to RSVP is on that slide, and I'm also going to put it in the chat. If you want to just spend your entire day with us, you can join me and Judy from the public library for the event that we're putting on for her community right after this one. So let's get that link in that chat. Globe Clouds Library. So if you all have any questions, feel free to get those in. And someone said, will this program from the start be available afterwards? It's already available, actually. We've been streaming live to YouTube this whole time. I'm also going to email it out after, and it'll go on that scistarter.org/NLM page we've been pointing you to, so not to worry. So Eterna, this is one of my favorite projects because I think it's so great for our virtual environment, especially, let's say we can't gather in person, right? But we still want to engage a group in a shared project. Eterna, it could be a perfect fit for your library and CBO. I'm going to let Doosoon speak a little bit on that, playing in groups and just having fun with your community. So Doosoon, over to you.
Yeah, thanks. So two things that we've really focused on, especially in light of the shutdowns that have happened in the COVID-19 pandemic, are how to create smaller communities such that starting out doesn't seem as intimidating as jumping into a pool of thousands of players. So what I've shared on the bottom right is a screenshot of what our groups page looks like. I remember, I think, seeing that someone was tuning in from Qatar. So we actually have a university team out of Saudi Arabia who is on Eterna and participating in research through a team from KAU there. And we've built out a lot of different ways to break out into small communities. So you could, in theory, have a team from your library, wherever you may be tuning in from. So we've focused a lot on sort of onboarding new users and making sure it's really friendly for new players to join Eterna, especially during the shutdown. And then the second thing I briefly touched on earlier was our education initiatives that we've launched to integrate Eterna into your scientific curricula. So if you're a librarian and you're looking for a fun way to get more students, early students involved in science, Eterna could teach a lot of the very basic RNA biochemistry or synthetic biology or molecular biology that might really, in a fun way, supplement sort of a formal scientific education that you might see. So we're working on things, we're open to more feedback and working with community leaders like librarians or small group leaders to make Eterna to make the platform more accessible to as many people as we can. I see that we have a question from one of the audience members, how long it's been around. So it started in 2010, I believe. So we've been around almost about 10 years, going on 11.
And actually, I'll ask a quick follow-up on that. How has Eterna changed since you first got involved?
Oh my gosh, I think the first bit is how much technology has changed when Eterna was launched. Like the things that you can do on websites now are just vastly different. So we're able to reach way more people. I think the biggest thing that has changed is the depth with which people, especially the longtime players, dive into on Eterna. So Eterna went from, here's a fun way to spend a couple hours on my couch playing a game, which if that's what you'd like, that is still available. But now we have players who are really interested in, when are we getting a publication out? Can you send me scientific papers and help me understand them? And there's a whole team of scientists on Eterna whose job it is and who are excited to do that for citizen scientists, is to really give them a sort of let's say, an informal graduate education in RNA biochemistry by sending them scientific papers, by explaining to them the concepts that are inside of them, and also sort of pushing the boundaries of the depth that citizen scientists are wanting to go to.
That is amazing. And we'll keep these questions coming, everybody. While you get your questions in the chat, I wanted to let everyone know Eterna is a Scistarter affiliate project, meaning that if you're a Scistarter user, you can actually track the number and frequency of your contributions in your Scistarter dashboard. And this is particularly relevant for libraries, CBOs, and educators, because let's say you make a Scistarter list. And you can find more info about Scistarter list at the bottom of that NLM page, so scistarter.org/NLM. But basically, what that list is it's a way to share projects and special instructions with your community. It gives you like a public-facing web page on Scistarter, and then also to access analytics about the impact of everyone you shared that list with. You'd be able to see, oh my gosh, there have been 300 contributions to Eterna since I shared this list by people who have viewed this list. So that's been helpful. Like if you're a part of a library staff, you may need to put together some statistics about your impact. That might be helpful to you. So feel free to go to the NLM page to get that. We also have a question in the chat for due soon. Jasmine wants to know, are the publications out of this platform published open access?
Yes, that's one of the big things that we're pushing to make sure that all our publications are openly available. So on the Eterna website, there's a publications page that lists everything. And in the case that a journal article is not open access, members of the Eterna community will make sure that you have access to them if you're an Eterna player.
That's awesome. And I have a question. I think this one's for me from Brenda in the chat. She wants to know, how many citizen science webinars have there been in 2020? And also, are they recorded to be viewed again? Yes, Brenda, we have. So I've personally facilitated well over 100 webinars this year, but there have been so many. I know like Due Soon, for example, he's been doing those weekly town halls, which I think you could probably think of as webinars in a way. But if you want to watch more citizen science content, go to our Scistarter citizen science videos page, scistarter.org/citizen-science-videos. But I think I'll pivot this into a question for Due Soon. What have those town halls been like? How have they, evolved throughout this time? And what do you all talk about each week on those town halls?
It varies widely. So I actually started the town halls when I joined the platform as a way to directly talk to citizen scientists myself to get an idea of where we should focus our efforts on Eterna. So in the very beginning, it was me asking a lot of questions to the citizen scientists, asking, here's the scientific challenge. Do you think we can take it on? What do we need to develop for you so that you can be best equipped to address this research problem? From there, it turned into, they had a lot of questions for me, a lot like the introduction slides you might have seen on mRNA therapeutics. How do mRNA vaccines work? Why do we need to redesign them? What is some literature out there that might help us get a head start, at least scientifically, on what to design for? All the way to nowadays, especially, we've had 7 rounds of the Open Vaccine Challenge where they give us a design, we test them, we return the data. So lately, it's a lot of here is the data that's coming out, and I'm going to explain it to you and take questions, of course, and it's always a discussion, what the data mean and what I think the takeaways are. And often players will say, well, actually, can you format the data in this way and send it to me? Like, I want to mess around with it myself instead of you doing all the analysis. So lately, it turned out a lot into a more in-depth conversation about the data that are coming out. Often, we also get, we have guest speakers on the town hall. So, 1 week, I think the citizen scientists were really interested in intellectual property and what that looks like when citizen scientists develop a medicine. What might that look like in translating that to a medicine that can be commercialized and given out? So we invited a professor of law from Penn State to talk to us about how intellectual property is handled and how different Eterna is compared to other platforms that are used for discovering medicines, which are usually kept behind closed doors at big pharmaceutical companies. So it varies widely. I would say the agenda is set more by the players than it is the developers or scientists. And we just have to come in ready to answer a lot of questions and ask a lot of questions ourselves.
That's amazing. I love that. I know we had promised that we would address the concerns of educators a little bit more in this breakout group. So if you don't mind, Dusun, I'd love if you could kind of speak broadly to that educator-focused audience.
Oh, sure. Sorry. What
So educators they just wanted to get a little bit of advice from you about incorporating Eterna into their programming efforts.
Oh gosh, I think I would venture to say that the educators would know much more than me how to do it. But I think the biggest sort of tip that I would say is just to reach out. A lot of these efforts that we have taken on the Eterna have come from what many would consider a cold email to me or to one of the other developers on Eterna. And I would say it's a smaller team that you might imagine. So if one of our team members gets an exciting email from an educator, that very next week at the developers meeting, we're probably talking about it and asking ourselves, like, how can we be more involved with that initiative? Or like, this is a really exciting group of people we want to work with. How can we make that happen? So the first step would be just to reach out and see if we can tailor something for you. The second bit towards educators is just to, I don't know, keep doing what you're doing, because you know your audience the best. Like, I would never go into a classroom and tell a teacher how to teach her or his students, right? Like, I think educators have a really, really unique and valuable insight into their audience that people from, let's say, my end, the platform end, should listen to more and help cater our content to what you need.
Definitely.
So I would say, take more initiative in asking for things, because those things are invaluable to us in sort of us allowing to better our platform to integrate better with education-focused initiatives.
That's amazing. Yeah. And I also wanted to quickly speak to the education audience specifically. There may be interesting tie-ins. So something I've seen is, like, of course, this is a natural fit for a class where you're studying mRNA, right? But if you're an instructor of English, for example, you can assign your students Eterna to write about and teach them science writing. I've seen educators do that even at the college level for introduction to composition classes. So I've just been in awe of, like, the creativity of different educators. I think that's so amazing. And if you all have a particular success story, as you use this in your classroom or in your library, in your community-based organization, we're always happy to highlight those. Tag Scistarter on social media, Eterna on social media, and also shoot us an email. But we have a question in the chat, another one for Dusun. Can you address the issue of vaccine safety? This issue seems to be a concern, causing pause for people to receive a vaccination.
So I can say personally, I am incredibly pro-vaccine, because if you look at the history of epidemiology, which studies diseases at the population level, vaccines probably have saved more lives than any other technology medically since inception. Now, I think a lot of the concern that might be coming up is because there's all this talk about how quickly we develop these vaccines. And people might be worried about their safety and efficacy because of how quickly it was developed. And I think this steps away from the scientific question a bit, because it has to do with, do you trust the FDA to have done the due diligence to make sure the vaccine was safe and efficacious? And the data has been publicly released to the public, and a lot of professional scientists have combed through the data to make sure it's rigorous. And what I've read and what many professionals have agreed upon is that the data, which ultimately determine at the ground truth level whether the vaccine is safe or efficacious, all the data point towards the vaccine being effective, and all the data certainly points to the vaccines being safe. So I would say from a safety and efficacy perspective, you have nothing to worry about, because this vaccine was developed faster, because the FDA, it seems, did not lower their bar for the amount of data and the scientific conclusions that have to be drawn for this vaccine to be a good vaccine. The only part that people admit that they are maybe unsure about are questions that can only be answered with time. Things like, if you take a vaccine for COVID-19, how long does that immunity last? right? That's a question you can't answer until you can literally wait that amount of time. So I believe the FDA recommendation clearly states that that's possibly a limitation and that's something they have to wait on. So I believe the recommendation would be to get the vaccine to give yourself the best chance of fighting an infection if you become infected, but keep taking those precautions until we have more data that present otherwise. So I think people for scientific questions like this one, I prefer to always turn to the data to let them speak for themselves. And I think that's what's so wonderful about science, is that science relies on a set of ground truths and not what it's reported and stuff like that. You can just always look to the data to see what the conclusions that you should draw are. So it was done faster, but it was not done in any sloppier way, because if it was, then it wouldn't have gone through in the way that it did. What might also be encouraging is that there are other vaccine candidates that are coming out, and although you might think you want those out on the market as fast as possible too, they're actually experiencing delays because they're finding that it's not as efficacious. So maybe it's an example of the FDA doing their job and saying, the data don't look as good as they should for these to be distributed, so you have to work on that before we approve it. So there are instances where the FDA is shooting down vaccine programs at certain stages until it meets their bar for efficacy, which I think is very encouraging, that they're approving some and negating some. That lets you know that there's a threshold that they're not compromising on.
Yeah, that's amazing. And that's really heartening just to hear. We have another question for you. Does Eterna only work with mRNA, or does it incorporate other RNA structures or DNA structures?
That's a fantastic question. So this is actually our first project that involves mRNA design. So every other project that I mentioned that we've taken on and we have thought about taking on have not been around mRNA. And this is maybe a good segue into the fact that not all RNA in your body is mRNA. There's a lot of, I would actually say there's more non-mRNA RNAs in your body. And we have focused almost exclusively on those other RNAs because they have really interesting structural problems that we are trying to solve. So we work on almost exclusively outside of the Open Vaccine Project, non-mRNA RNA molecules. We haven't explored DNA because it becomes a totally different set of scientific questions, and we right now don't have the expertise in those areas to work on it, but we certainly have the engineering capability to introduce some of those puzzles as well.
That's great. So everybody, please get your questions in. And while you do that, one thing that always helps me conceptualize these things, especially for our leadership breakout group, is to hear examples and anecdotes. Do you have any examples of I know you already talked a little bit about these examples earlier, but examples of success stories with libraries, community-based organizations, and educators, facilitating Eterna with their communities, and any best practices learned from those examples?
I think I have the most experience on the education side because we have worked this summer to try to really push Eterna to supplement science curricula. Mm-hmm. And the biggest thing I've learned is that the most valuable time is spent listening to the teachers and also directly speaking with the students. The best practices from our side that I've learned is to just keep my mouth shut and listen to see what the students have to say and what the teachers have to say, and trust that they more likely know what they are talking about more than you do. So our best ideas and our best initiatives have not come from us internally, but us just sitting down at a town hall and listening to educators suggest things. So the best practice for the educator side would be to speak up more often and to suggest things that maybe seem silly to you, or insights that you have and it's so obvious to you because you've been an educator or a community leader for a long time. But to us, it's just like a huge revelation. Right. So I think recognizing your expertise and the insights you have in your communities and speaking out for those are incredibly valuable for us. And I think it's for the benefit of the audience members, like the students or library members, to do so. So my biggest best practice is to just listen more to for lack of a better word, the customers, the people who are going to use your platforms.
Yeah, and this is last chance for questions, everybody. Get those in the chat or the Q&A. And for the other members of the panel, like Nancy, Michael, or Emily, if you have any other questions, feel free to unmute. And while you guys generate those in your head, I wanted to ask you soon, is there anything else about Eterna that you haven't mentioned yet that you'd like to tell an audience of community leaders?
Anything else about Eterna? Gosh, what have I? There's so much, and maybe one thing I wanted to point out was that Eterna is a community that's about more than just designing RNA molecules. So one person had a great question about how to use the data on Eterna to teach students data analysis. We also have philosophers on Eterna who think a lot about the social implications of citizen science and developing medicines through a democratized way. So even if your interest isn't specifically scientific, so let's say you're a librarian and you're not necessarily interested in a very science-heavy activity, but you're just interested in the community of like how citizen science might be used in medicines, and you have questions surrounding the topic that aren't just designing RNA molecules, I would say we welcome and we really are privileged to have members like that in our community. So to sort of jump in and if your interest is not becoming the best RNA designer, that's totally fine. But you might be the person that raises the right questions about ethics or the philosophy behind science. That might be incredibly valuable for us, and we welcome that input from different perspectives.
That is great. Any last minute questions from the panel? And if not, I wanted to point you all back to the scistarter.org/NLM page. You'll find instructions for Eternas there, more tips about facilitating citizen science in groups and in your community, and the Library and Community Guide to Citizen Science and other recordings from the Citizen Science and Library series. And that being said, just a big thank you to Dusun for the work he does and for educating us today, and to all of our partners, especially NNLM and all of us. And I'm going to pass the mic to Nancy to finish off our event and talk about how people can get their MLACE credit if they're so inclined. Over to Nancy.
Thanks, Caroline. So before you all go, thanks for all your questions and participation in this webinar. A special thanks goes to our guest speaker, Dusun Kim, for an excellent presentation. This is our last webinar in the Citizen Science and Libraries webinar series, and the link to the webinar evaluation will be provided now in the chat box. So I'll just put that in there. For those that want MLACE, the evaluation will have an MLACE code for you to use to get your MLACE credit once you fill out the webinar evaluation. Any questions that you have about the MLACE can be directed to me at nkshin1@uw.edu. Thanks again for your participation, and thanks again to Dusun for a wonderful presentation.
All right, well, I think we'll end it here. If you all want to keep on spending your day with me, feel free to join me for the Globe Clouds program. I'll put the link in the chat for that again. And otherwise, I'm sad this is the last one in the series, but I'm glad that we're coming back strong in January with more programs with NNLM and more fun on our scistarter.org/NLM page. So I put the link in the chat for the Globe Clouds program. Oh my gosh, I keep on going too fast. I'm also going to put the link in the chat for the NLM page where you can monitor that page for more programs, especially ones for libraries and CBOs. And have a great day, everybody. Thank you so much.